So after testing the microphone, we're going to officially start this DIM Symposium. So that's the last one of this first mandate of DIM ELICIT. So we started this back in 2016, submitting the application with Patrick Tabling and Jean-Christophe Oliveau-Marin, who cannot attend today. We had the opportunity to develop this network and to fund projects in the field of technologies for life sciences to make an impact, deliver novel technology that can drastically change how you will measure, investigate biological phenomena, and those in the field of microfluidics, photonics, waves, and image analysis in the first place. So today you will have a number of talks of scientists presenting what they've been doing in a project funded by the DIM in the different fields. Well, I'm sure it's going to be super interesting. So I want to really thank the Région Île-de-France for letting us the possibility to develop this scientific topic in the Paris area, and to bring together scientists, both technologists and biologists, to develop this new type of tools. So Marina Caillet, our superior project manager, will make a short presentation of like key figures of this first mandate so that you can have an idea of the number of projects and some key result of this program. And then we're going to also briefly in this introduction give you some information about the new submission that we made on your behalf of this community to Région Île-de-France to renew the program. So Marina, the floor is yours.
Donc, bonjour à tous. Merci Elodie pour cette introduction. J'essaie juste de régler le micro. Voilà, je pense que tout le monde m'entend bien. Donc, le DIM ELICIT, c'est le domaine d'intérêt majeur Empowering Life Science with Innovative Technology, ou renforcer les sciences de la vie avec les innovations technologiques. Aujourd'hui, le DIM ELICIT, financé par la région Île-de-France, c'est un consortium pluridisciplinaire qui compte plus de 700 chercheurs. Co-coordonné par l'Institut Pierre-Gilles de Gennes et l'Institut Pasteur, le consortium s'est co-construit autour de 9 partenaires fondateurs. Mais bien entendu, il a toujours été ouvert à l'ensemble de la communauté francilienne, de toutes les équipes qui sont impliquées dans les thématiques du DIM ELICIT. Cette volonté d'ouverture forte et propre à la coordination, elle est directement liée aux objectifs du programme. Le premier objectif, c'est effectivement de créer une communauté entre technologues et biologistes. Le second objectif est justement de financer et de créer de la valeur scientifique à travers le financement de projets de recherche, mais aussi projets de recherche ambitieux et innovants. Et enfin, le dernier objectif est directement lié aux besoins de la région Île-de-France, à savoir de créer de la valeur socio-économique à travers le soutien des startups, mais aussi de la propriété intellectuelle ou encore de la formation des jeunes chercheurs. Comme on peut le voir sur cette diapositive, le DIM ELICIT s'articule au niveau de 3 domaines thématiques, à savoir la microfluidique, la biophotonique et ondes, l'analyse d'images et le big data. En plus, on retrouve 4 axes applicatifs qui sont la cellule unique et la molécule unique, l'organe sur puce, les technologies pour l'étude in vivo de modèles animaux et enfin les technologies performantes et à bas coût. À travers le DIM ELICIT et grâce à une subvention de plus de 11 millions d'euros, c'est plus de 80 projets de recherche collaboratifs qui ont été soutenus depuis 2017. On distingue 2 grandes catégories de projets au sein du DIM ELICIT, à savoir les projets dits de co-développement qui peuvent faire émerger des technologies innovantes et les projets de dissémination d'une technologie vers la biologie. Dans les deux cas, il s'agit d'une collaboration entre technologues et biologistes. Comme on peut le voir sur cette diapositive, dans les deux types de projets, il peut y avoir l'intervention d'un partenaire industriel. Et dans les deux cas, les projets peuvent conduire à la création d'une start-up. Si on s'intéresse à la répartition des projets entre 2017 et 2020, on peut voir que le DIM ELICIT a financé 20% de projets de dissémination et 80% de projets de co-développement. Pour pouvoir illustrer justement la philosophie du DIM à travers ces idées de co-développement et de dissémination, je vais vous présenter très brièvement deux exemples de projets. Mais l'ensemble des projets qui vont être exposés au cours de la journée reflètent justement cette idée de co-développement et dissémination. Le premier exemple est un projet de co-développement qui rassemble l'équipe de Cathy Ventalon, technologue, avec une équipe du Collège de France, Michael Zougaro, pour la partie biologiste. Ce projet a été subventionné en 2017 par le DIM ELICIT et consistait au développement de systèmes optiques permettant d'observer à l'échelle cellulaire et in vivo l'activité de l'hippocampe du cortex préfrontal chez le modèle murin. Grâce à ce financement du DIM ELICIT, les deux équipes ont pu ensuite soulever d'autres financements. Ils ont aussi publié des articles scientifiques et participé à plusieurs communications scientifiques. Mais surtout, ils ont pu, grâce à un partenariat aussi avec deux entreprises privées, la société Imagine Optique et la société Cartala, et le soutien de la cellule de valorisation de PSL, monter une start-up en 2020, la société Imetry. Autre exemple, pour un projet cette fois-ci de dissémination, qui consiste au développement d'une plateforme de microfluidique pour la culture en cellules 3D, avec une subvention du DIM ELICIT en 2018, sur un projet porté par Charles Barou, qui a donné lieu à la création d'une start-up en 2020. Je n'en dirai pas beaucoup plus sur ce projet, puisque Charles va nous présenter ses résultats en fin de matinée. Comme nous l'avons dit, à travers le DIM ELICIT, c'est plus de 80 projets collaboratifs qui ont été financés, et notamment plus de 40 équipements financés en Île-de-France. Comme on peut le voir sur cette infographie, on a la représentation de la localisation des équipements financés à travers le DIM ELICIT et la répartition selon la thématique de recherche associée. Comme on peut le voir, dans le 5e arrondissement de Paris, où nous sommes actuellement aujourd'hui, il y a plusieurs instituts de recherche qui ont été financés, notamment l'Institut La Vision, l'IPGG, mais aussi le SPCI ou encore l'ENS. Si on prend l'exemple de l'ENS, en bas de cette diapositive, à la pointe, on peut voir qu'il y a eu un projet de microfluidique qui a été subventionné pour un équipement et 2 projets de biophotonique pour des équipements. Comme je l'ai précisé, les financements des projets de recherche au sein du DIM ELICIT peuvent parfois donner lieu à des partenaires avec des partenaires socio-économiques, des startups, des SME, voire même des PME. Depuis sa labellisation en 2017, c'est plus de 20 partenariats qui ont été mis en place dans le cadre des projets du DIM ELICIT, et c'est ce qui est représenté sur cette diapositive. Nous pouvons voir à chaque partenariat le logo de l'institut académique avec le logo de l'entreprise et le nom du porteur de projet. Par exemple, vous pouvez voir que pour les projets de 2018, il y a eu le SPCI a une subvention du DIM ELICIT en partenariat avec la société Platon pour un projet porté par Mathilde Ressa. Autre exemple, en 2019, en bas de la diapositive, vous pourrez voir que l'Institut de l'audition a eu une subvention du DIM ELICIT en partenariat avec la société Cartala pour un projet porté par Brice Batelier qui nous présentera ses résultats cet après-midi. Autre point, au sein de DIM ELICIT, en plus des partenariats industriels, il est possible aussi que le projet aboutisse à la création de start-up. Depuis sa labellisation, c'est 5 startups qui ont été créées en lien avec des projets financés par le DIM ELICIT. Nous avons vu dans les 2 exemples que je vous ai présentés, la société Imetry, mais aussi la société Ocomera. Mais il y a aussi la société Avatar Médical, qui est soutenue justement par l'Institut Curie, PSL et l'Institut Pasteur, grâce à un projet porté par Jean-Baptiste Masson. Il y a aussi les sociétés Everzoom et Evora Bioscience, qui résultent de projets subventionnés par le DIM ELICIT en 2018 et portés par Amanda Silva-Brun, qui nous présentera ses résultats cet après-midi. Enfin, à travers le programme du DIM ELICIT, c'est aussi plusieurs manifestations scientifiques qui ont été soutenues. Comme on peut voir ici représenté sur cette diapositive, entre 2018 et 2021, c'est plusieurs manifestations scientifiques qui ont été financées par le DIM ELICIT. Aussi bien des manifestations scientifiques destinées directement à un public de chercheurs que des enseignements ou encore des concours comme le In Contest, où le DIM ELICIT a soutenu un groupe d'étudiants pour participer au concours et déposer le projet Combine, qui a été sélectionné et a obtenu une médaille d'or. Le succès du DIM ELICIT ne tient pas seulement à la qualité des projets de recherche et au caractère innovant, mais il tient aussi à une animation forte de son réseau. Et cette animation passe avant tout par un site internet créé depuis 2019 qui est dynamique et qui est enrichi régulièrement avec de nouveaux contenus. Pour assurer justement le lien entre le réseau et notre communauté, il a été aussi créé une newsletter qui compte aujourd'hui plus de 600 abonnés. Le DIM ELICIT est aussi très actif sur les réseaux sociaux avec un compte Twitter, un compte LinkedIn et une chaîne YouTube. Cette chaîne YouTube est justement alimentée avec des vidéos plutôt destinées grand public en partenariat avec l'Esprit Sorcier. Et j'en profite pour remercier tous ceux qui se sont prêtés au jeu du film avec l'Esprit Sorcier. En plus du soutien à l'animation et au dynamisme de notre réseau, l'animation passe aussi par le soutien à la formation, notamment la formation aux entrepreneurs avec le Deep Tech Founder dont vous entendrez parler à la fin de cette matinée. Il y a aussi le soutien à la formation, à la communication, notamment avec la participation du DIM ELICIT avec d'autres DIM au cycle de formation inter-DIM en partenariat avec l'association Trace. En parallèle, bien entendu, il y a aussi les événements organisés directement par le DIM ELICIT. Donc, par exemple, le DIM ELICIT Day en 2019 ou encore le DIM Covid, spécial Covid réalisé exclusivement en ligne au début d'année. Et aujourd'hui, bien entendu, le colloque auquel vous participez. En plus des actions directement liées à la communauté du DIM ELICIT, la coordination a toujours voulu et s'est rapidement engagée dans des actions de médiation scientifique, donc cette fois-ci destinées au grand public et au public scolaire, notamment avec l'organisation de l'opération Sciences pour tous, destinée aux des groupes de 3e, mais aussi en participant à des actions inter-DIM, notamment au niveau de la Fête de la Science en 2019 et en 2020, où il a fallu tous se réorganiser pour pouvoir faire des événements exclusivement en ligne. Donc ça a été le cas avec la création d'un escape game. Enfin, il y a aussi des domaines un peu plus nouveaux avec le partenariat Savant Sachant Chercher pour la réalisation de podcasts. Et à présent, je vais laisser la parole à Élodie pour parler de la suite. Merci à tous.
Merci, Marina. Thank you. So switching back to English, because I think it's important any scientist gets what's happening with the renewal of the program. So basically, the new DIM ELICIT, now it's going to be DIM ELICIT, domaine de recherche et d'innovation majeure, but we are not changing the rest of the name, because essentially it's more of the same with some twists that I'm going to explain now. So first of all, we were 3 coordinators, Patrick Tabling, Jean-Christophe Oliveau-Marin, and myself. And for the new program, Patrick decided to pass it to Jean Baudry from ESPCI here. So Jean is going to replace Patrick Tabling as one of the 3 co-directors. So here, you know, I want to take the opportunity to thank you, Patrick, for doing this with Jean-Christophe and myself over these past 5 years. I mean, it was a great pleasure to work together, and he did a lot to make it success in the first place 5 years ago in front of the jury of the region. So we should be thankful. And so, yeah, essentially the core of the program stays the same, developing new technologies for life sciences, enabling at the end of the day, new instruments that are going to change the way research in biology is done, and collaboration between technologists and biologists to do so. That remains the same. We're still going to finance the same kind of collaboration project, of dissemination projects, but we are a bit extending the fields, the scientific fields, which can have an impact to do so. That's, the primary reason for this is that during these 5 years, we've seen a number of proposals come to the DIM, which were not exactly fitting the scope of the scientific topic, microfluidics, biophotonics, image analysis, and especially in chemical biology, biochemistry, molecular biology, all these topics which can, at the end of the day, have an impact in the world of instruments for life sciences. So, take the example of the Illumina technology. Okay, it's a machine, it's microfluidics, it's advanced image analysis, but it's also in the very first place, the biochemical reagents which are going to generate the signal. So we are adding this chemical biology axis to the program, and also new steering committee member to support it in the scientific steering committee, Arnaud Gauthier from Sorbonne Université and David Bicard from Institut Pasteur. So that's one novelty. Regarding the application axis, we also decided to let them be a little bit more applied, because some of this technology for life sciences, at the end of the day, they can become new in vitro diagnostic technology, and that's where they're going to make an impact, or they can become medical technologies, and for instance, like new medical imaging technologies. And we decided that this would also be part of the program, provided we stay at the cellular scale, tissue scale, so the biological scale, and not like the human medical imaging or like more behavioral diagnostics scale. So these are the main changes about the scientific axis of the program, but you can say it's largely staying the same. We also continue what we have developed in supporting collaboration with the industry, and trying to have industrial partners take part in the program, trying also to promote and help the creation of startups, like continue to finance trainings for scientists who are interested to receive them, or networks of scientists which have created companies that can talk to scientists who want to create companies. So we're going to continue down that road, and also with all this dissemination to the public activities that we started in the very first place. So the dossier was submitted last week. So this was like a big chunk of work for September. So now we're waiting for feedback from the region, and this is going to happen at the end of this year. So of course we will keep you, our community, posted in the first place about the results. So if you have any question, that might be the moment to ask them about the renewal of the program, or you can catch us later, Jean, myself, to discuss about it. And so, and if not, then we can move on straight forwardly to the scientific talks. Okay, other question on the Zoom, maybe? Discussion like there. Okay, this was just about the camera, not displaying the speaker. So if you're fine, then we're gonna move to the first talk.
Je ne peux pas quitter l'écran. comment? Quitter l'écran. Quitter l'écran. C'est ça. C'est bon?
Merci beaucoup. Okay, so hello everyone. I'm very happy to be here, and I would like to thank first the organizers for giving us the opportunity to present you these new concepts in terms of super resolution imaging and the way it could be applied to improve our knowledge on a biological topic, which is regulation of microtubule dynamics. So I will first present the biological part, and then Sandrine will take the floor to give you explanations about the technology.
I'm sorry. On the Zoom, we cannot see the screen. Can you share the screen, please, on your computer?
Le partage d'écran, c'est pas moi qui gère. Is it okay now?
No.
No. Oh. Oui, c'est le deuxième fichier. Je peux pas enchaîner derrière si tu veux.
So it works now. Thank you.
Okay, thanks. Hopefully it will be okay till the end. Okay, so just to remind you maybe about microtubules, which represent a dense network in cells, which is essential to organize the cytoplasm, cell polarity, for example, and also highly involved, of course, in allowing cells to divide by building the mitotic apparatus. And these microtubules are made of the assembly of tubulin dimers made of an alpha and a beta subunit, which combine to GDP or GTP. And the assembly of these tubulin dimers build a hollow tube of about 25 nanometers in diameter, which is made of the juxtaposition of about 13 protofilaments, one protofilament being a linear assembly of tubulin dimers always in the same direction. And the extremity of the microtubule that exposes beta tubulin is called the plus end. And this plus end is generally found at the cell periphery, and it's the place where microtubules exchange subunits with the soluble part of the cytoplasm. And if one records the behavior of microtubule as a function of time, then you can see the alternation of phases of growth and shrinkage, and transitions from growth to shrinkage, for example, here are called catastrophes, where the reverse, I mean, the interruption of shrinkage and the resumption of growth is termed a rescue. So these make microtubules very dynamic and allow them to explore the cytoplasm to fulfill their function. So from a molecular point of view, the elongation of a microtubule results from the addition of a GTP-bound tubulin at the plus end of a protofilament and the creation of a new interface here between the new and the old dimer will stimulate the GTPase activity, which is held by the beta subunit, which will hydrolyze GTP into GDP plus phosphate. And this hydrolysis, therefore, mean that most of the microtubule body is made of GDP-bound tubulin, with the exception of the last incorporated subunits, which are made of GTP tubulin, and therefore represent what's called a GTP cap. The loss of this GTP cap will cause microtubules to depolymerize by bending their protofilaments outwards and releasing GDP tubulin. So the textbook model for microtubule dynamic instability, sorry, I think something weird happened with the sharing. Is it okay? Yeah, okay, thanks. So the textbook model about microtubule dynamic instability says that it's the loss of the GTP cap that causes catastrophes, and of course the recovery of the GTP cap will promote rescue, but this mechanism has long been elusive. And some years ago, together with Frank Perez in the Institut Curie just near here, we tackled this question by selecting recombinant antibodies against GTP-locked conformation of tubulin. Like this, and the obtention of several clones, and especially the one that's called MB11 here, allowed us to label GTP caps for the first time in cells. And you can see, for example, on this zoomed area, that there are some microtubules which actually display a positive signal here in red at the end, at the plus end of microtubules. But also we were surprised by the presence of some internal signal in the form of these dots that we call GTP islands. And strikingly, the presence of GTP islands could be very well correlated with the location of rescues, because if we record dynamic behavior of microtubule in this sequence, then identify the locations of the rescues with these colored arrows. The retrospective labeling of the microtubules shows that GTP islands are actually present at the locations where the rescues occurred, and these were not scars, but really cause of rescue processes. So to go a bit further, to get still with Frank and with Anne-Marie Aguirre's lab in Marcoussis, we also analyzed the distribution of these GTP islands in cells. And strikingly, the distribution is not stochastic, but about half of the GTP islands co-localize with microtubule crossings, just like the occurrence of microtubule rescues, which also occur at about 50% at crossings. So microtubule crossings happen to involve, of course, a crosser microtubule and a crossee, and the occurrence of the GTP islands can happen in crossers as well as in crossees. To give you an example of what happens in crossers, you can see this blue microtubule here that grows and crosses the yellow one at this location. And after the retrospective labeling, there is the presence of a GTP island exactly here. We also addressed the question by in vitro experiments. And in these in vitro experiments, we took advantage of the possibility of microtubules to bind strongly to molecular motors. And the growth of these microtubules on a cover slip covered with recombinant molecular motors that were blocked in terms of motility allowed planar constraint to microtubule growth. Therefore, they grow and then they have to collide when they cross. And either the performance of these microtubule collisions, but also microtubule sedimentation over natural or artificial nanoreliefs, I mean other microtubules or artificial pillars, all made Gtp islands to occur. So this happens in regions where microtubules are mechanically constrained. So what could be the nature of these Gtp islands? The first possibility is the presence of lattice defects. For some reason, one of the microtubules will be damaged at the crossing, which could also cause self-repair. There could be also a shift in the number of protofilaments. And the second possibility is also the presence of local bending. And I will briefly discuss at the end of my talk about the role of tubulin acetylation in this context. So first, microtubule damage and repair. It's quite well known now for a few years that mechanical stress, repeated bending, for example, or laser photo damage, as shown in Manuel Terry's lab, or nanoscale damage by severing enzymes, catenin or spastin, or nanoscale damage by the work also of molecular motors along microtubules. All these can cause microtubules to damage and to self-repair by incorporating new tubulin subunits, which is frequently the cause of rescues. And in the context of our studies, we also identified that the repaired regions that happen at microtubule crossings often coincide also with the presence of Gtp islands. So the next possibility was a shift in the number of protofilaments. It was possible in vitro to perform experiments where microtubules are annealed end-to-end. And we took advantage of this using either Taxol stabilized microtubules, which display 12 or 13 protofilaments, and microtubules assembled in the presence of a Gtp analog called GMP-CPP, which display 14 protofilaments. So by performing annealing between these two types of microtubules, we could get single microtubules with lattice defects and shift in the number of protofilaments at predictable location if we use microtubules of different colors. So this is what is illustrated here. The annealing of GMP-CPP microtubules in blue with Taxol assembled microtubule in green. All display frequent labeling of Gtp islands at the microtubule interfaces. Of course, this is the control with two GMP-CPP microtubules. So to summarize this, the microtubule defects and disruptions can result from a variety of events. And the consequence in the microtubule lattice will be architectural defects with voids, with a shift in the number of protofilaments, also possibly with twists in the helix of the microtubules. And all these voids can be at least partly filled by the recruitment of new tubulin subunits, which can therefore incorporate into the lattice and be recognized by the antibody. One should not forget also that what happens in cells is, of course, dependent on the architecture of the microtubule, but also on the presence of associated proteins. And there are some proteins like this one, which is called CLIP170, which are rescue factors. And we showed recently that this protein can not only bind to the tip of growing microtubules, which is known for years, but also that it can interact with these microtubule disruptions. This can be observed in this short video where you can see several microtubule comets that mark the growing ends. And if you take care and look carefully at what's pointed in the bottom video, you can see that these comets leave trailing dots transiently along the microtubule, and these trailing dots actually colocalize with the presence of Gtp Islands. And recently, we proposed that the transient retention of CLIP-170 at Gtp Islands could be a way for cells to boost microtubule repair and to prepare the lattice for future rescues. To finish, I would like to switch on the third possibility I presented, that is local microtubule bending and curvature. It has been striking for years that some microtubules in cells, especially those that are highly stabilized, that resist depolymerizing treatments like here in the presence of nocodazole, these microtubules, these resistant and stable microtubules are very curly, which is quite counterintuitive with the architecture of a microtubule, which are hollow tubes and could display a high flexural rigidity. Okay, but these highly stable microtubules actually are highly modified. Tubulin is especially hyperacetylated due to the activity of an enzyme called tubulin acetyltransferase, or TAT, which is expected to enter the microtubule lumen and then to acetylate alpha-tubulin inside the lumen of the tube. And in these experiments, it has been shown clearly that in the absence of that, either in cells or in vitro experiments, microtubules lose their flexibility and then more frequently break at mechanical constraints. So the present model postulates that if a microtubule bends, then some opening will occur in the lattice, which allows the entry of that into the microtubule lumen and then the acetylation of tubulin locally. This local acetylation of tubulin will loosen the interactions, the lateral interactions between protofilaments, which will glide more easily one with respect to each other. But this model could also perhaps be challenged because we've got some observations in the lab showing that upon cell stress, for example, after cell exposure to excess sodium chloride, there is a boost in tubulin acetylation within minutes. And this boost in tubulin acetylation is also accompanied by the high level of TAT recruitment onto microtubules, but also, oh, sorry for that, but also for an exchange between the unstressed and the stressed situation, because there is a short isoform of TAT which is present in basal conditions along microtubules, which is released here, while a long isoform is massively recruited instead. So this suggests that maybe the TAT localization is not only luminal. So to summarize, the questions we asked are, which is the nature of local disruptions? Can TAT localization be only luminal? And to address these kind of questions, of course, we need some improved super resolution imaging, which will be effective in 3 dimensions and have a resolution which is close to the size of the tubulin dimer, I mean 8 nanometers. And this will, I hope in the future, allow to better understand what's important in terms of microtubule network plasticity, and also the implications in terms of multiscale regulation, because one fascinating question is how the density of the microtubule network will also trigger the occurrence of many microtubule crossings, and this will influence at both scales, at the cell scale and at the microtubule scale, the control of dynamics. So before giving the floor to Sandrine, I would like to acknowledge the people from the lab who are listed here, and our collaborators from Institut Curie, from the LPN in Marcoussis, and of course Sandrine. and additional funding resources. And I switch to Sandrine.
Alors, elle est où, la présentation?
Sorry.
Thank you, Christian. So good morning, everyone, and thank you for giving us the opportunity to present the work we develop in collaboration. So the idea of this work was to find a new way to localize the single molecule in 3D. And if I quickly remind you, so initially, this is the kind of image we can get with Christian and tubulin, which is diffraction limited because all the molecules emit at the same times. And thanks to all these different strategies using either fluorescent protein or organic dye that we force to not all emit at the same time. So then we have all these molecules that emit Y by 1 in the point spread function, and we can localize them with a precision that strongly depends in particular to the number of photons they are emitted. So these approaches allow to go much beyond the diffraction limit, but in the first attempt, oops, sorry, it get a bit messy in the by coming on this computer. It's only a 2D. This localization principle only improve in 2D. Okay, so then you still need to know where is your molecule within the actual direction. So it means that you need to apply a complementary strategy to extract also the actual information. So among all these techniques, you have all these point spread function engineering when you kind of introduce some actual dissymmetries with astigmatism or double helix to know where is the position in Z. Of course, you have the supercritical angle that we somehow pioneer and leads to the creation of abelites. The problem with all these techniques is that they're only based on spatial analysis of the point spread function. And if we want to go much deeper, the main used techniques are all the point spread function techniques. So where you see that this astigmatism that is induced, if you look on the Kramer-Rao lower band that you can get, so the best precision that you will have depending on the actual position, you see that you always have a variation. So it's not uniform. And then it can really have, you can really get, even if you have a very strong astigmatism, you will have at least nearly a factor 2 between the position of the molecule in the focus and the one that are on the border of your capture depth. So it's not uniform at all. And furthermore, it both impact the lateral also information. So you degrade the lateral information by doing so. But the main problem is that when you want to go really in depth, which is the case we wanted to go with Christian. So not only looking at the first micron, but go 30 micron and even more. You see that you have the localization precision here, in particular along the axial direction, that drop very quickly because you can't really maintain this control aberration in depth, or you need to have some adaptive optics. So the whole idea of this work was, can we introduce a time parameter within the localization precision for the lateral and axial direction, and to see if it gets better in terms of localization precision. So what we all know is that when you excite your fluorescence samples with your laser, it will, if you have a low irradiance and you excite with a structure excitation like this, you will induce a modulated excitation. Okay, so the idea was, can we try to benefit of this modulation that you induce on your sample to extract the position of the molecule within the field of view? And in fact, that was the idea, is if we shift now this structure excitation along on the samples, what you would get is you will get, if you look at a point, you will see that it will have an irradiance that will vary along the time. So it will have a sinusoidal excitation and emission for each molecule within the field of view. So the idea is that if we now introduce this modulation, and if we stop here, for example, we can directly convert the phase information into an information on the spatial position. So if we stop here and we look at different molecules, for example, this one will be maximal and then get minimal. And this one, for example, will have a completely different phase because it starts close to a dark fringes and we have this kind of signal. So the difference of phase here when the modulation starts will directly tell you where is the molecule within the field of view. So of course you don't necessarily need to switch all the continuously the structure excitation along the field of view continuously, but you only need to extract at least 3 to 4 positions to extract the arctangent of the phase and get the position. So if you look back in the literature, in fact, this paper really inspires a lot. This was developed by the group of Jacques Pro in SPCI, where they were using this, they are creating interferences on their sample to follow, for example, these beads for myosin 5 stepping. And at this time, they already get a sub-diffraction, sub-nanometric resolution, but this was using scattering. And really, there was only a single event within the field of view. So the main challenge that we had when we think about using this kind of introducing a time modulation within the localization process was that we needed to not use scattering, but use fluorescence emission when the number of photons is much more limited. And we need to address a field of view where we have multiple emitters. And of course, we don't want only to do tracking, but do some structural imaging. So it means that it really changed a lot of things because it's not a photodiode anymore and a camera. So when we applied for this patent in 2016, we have all this in mind. And the main challenge we foreseen was to how we introduce this modulation, demodulation into something that is compatible with a camera-based detection. And then, interestingly, since then, there have been several, it's really active, oops, moving by itself, sorry. There is several groups working on this approach with different nicknames. And then some of the groups think it's come mainly from this development Miniflux, but they all forget that this, in fact, coming from this paper. So the idea is that now we have to replace, compared to a classical single molecule microscope, we have to replace the wide field excitation for structure excitation. So you can think to borrow any strategy that have been developed for SIM microscopy, so use either directly interference or gratings or digital micro device. And then you have to think how you're gonna demodulate this emission. So some of the groups want to use the camera directly to take the full image one after one. Okay, it keeps playing by itself, sorry. But then it's too low and then you lose too much event. So the idea was initially really to get the 4 different measurements of the intensity to sample the modulation within one camera frame. So then you can really free up and you can use a camera that can typically give you acquisition within 10 or 15 milliseconds, which might not be enough. And then you really demodulate within the same camera frame, sorry. So then for the excitation, so I will today just show you in the case of where we want to enhance the axial direction, but this concept can be translated into any direction. So but it means that you have to apply the structure excitation along the direction you want to improve. So for example, in the case of the axial direction, we keep the single objective configuration, and what we create, we create here a tilted pattern that is moved along this direction. Okay, so to do so, we split the laser in 2 arms, and then we have some electro-optic modulators that will shift the pattern on the sample. And here we have this knife edge mirror that allow you to choose where you focus the 2 beams here in the back focal plane on your objective to then create the fringage for the excitation. And then the electro-optic modulator shift that very fast, typically at kilohertz. So then you have to decide how to demodulate. So here for once, for those that more in physics, we use a Pockels cells at the detection. So they tend to be used for to build laser. But here we use a Pockels cells here that is placed in between a module that splits the image first here in two. Then the Pockels cells will have a given state of polarization, and the out image will then divide it again in two. So it means that we'll have four quadrants directly on the EMC-CD camera, and depending on the polarization of the Pockels cells, one quadrant will receive 50% of the light to sample the modulation. So all this is made in synchronization with a position of the excitation on the samples. And so you repeat this process 4 times. And each time you have a different quadrant for that is a privilege here on the camera. And then you get like this, the 4 samples in the 4 different quadrants that allow you then to extract the phase for each single molecule. So what is nice here with the Pockels cells elements is that it's really stable. There's no optical movement. It's not the fully optimal way to demodulate, but it's the most fastest and stable we've tested so far. So now, just a quick point on what is the precision you can get with this system. So this is the chromoral band that we have predicted, so around 6.5 nanometer for the actual resolution. And if we want to compare with which is predicted to be uniform, so this is the value you would get for classical astigmatism and extant astigmatism. So we see that you expect a gain that is at least between 3 to at least even 3 to 5. And then we did this precision measurement directly within a sample. So here, basically what we look at cells when we label with some dyes that are in the pen using some pent imaging. So it means that you can repeat the localization process several times and then extract what is the mean precision for this value. And you can see here for the actual precision that we look in the first 7 micron within the very same cells. And you can see that the precision is stay quite still, no matter the position in Z direction. And is like typically around 6.8 nanometers. What is also nice is that this very good precision that is uniform doesn't impact also the lateral precision that is also the same no matter the position in the Z position. Okay, so now of course, first things we tested with Christian was microtubules. And here, so the color coded is obtained thanks to this phase extraction, thanks to this modlock techniques. And now we can zoom in several parts. So on this 3D image, so this one was taken close to the cover slip in the first 600 nanometers. And if we zoom in this one, we can see that the microtubule is not really still and flat on the cover slip. So we can really see how it varies along the Z position here. And then we can zoom on these 2 small part of the microtubules, and you can see that we do have now the precision to see what Christian described, that there are some cylinder, so we can clearly see the hollow here in the center. So we also try on several other structures, like the clatrine here. So here it's at 4 micron above the cover slip, so a bit deeper in COS7 cells. And if you do this radial cut here, you can see that we do see that the clatrine coated pid here are organized on top of the nucleus. So they're reforming a shell on top of the cells. So this was done in this storm with Alexa 647. And you can zoom in here, and you can also do a small movie looking at these 2 clatrine coated pits. And you see that really like we do have a uniform precision because you see they're reforming this rounded shape. And then we investigated also some mitochondria. So this time at 6 micron on top of the cover slip. And we can also see that we have the precision to really see the bottom and the top part of the membrane of the mitochondria. And if we zoom in on this one here, you can really analyze, do some radial analysis here and see that you do localize all the molecule at the membrane here. and you can do some statistics on what is the size of the membrane you can measure, and you see that this is the same size on the bottom and on the top here, and we can repeat this kind of measurement to assess if there's any variation of precision here within the capture range, and you see that it's this thickness the same. And here this is a 3D movie on the very same mitochondria, where we can really see in 3D the structure. So here at 6 microns. So of course, having in mind the problematic we had with Christian, we wanted to go even much further. So that's where we started also to develop some kind of spheroid and organoid construction. So here we embedded in collagen matrix some cells where we labeled the microtubules. So here, this is the classical image. So you see that's the matrix is really strongly scattering. So this is the diffraction limited image where you see it's really like pretty blurry. And if you look at the single molecule event, you see that they are well distorted. So this would lead to pretty bad imaging typically. And here, because in fact, when we are doing the measurement for the axial positioning, we just sum up all the photons within the point spread function. So this is pretty, you can really extract very accurately the information to retrieve the axial position. So this is the image we can get in 3D here when we see here at the probably where is the central zone, I assume. And then you see that we have a very high dynamic here for the 3D, where the cells really like positioning like this. So just to conclude, so here this new technique, the MOLOG, so I've shown you only for the axial direction, but it could work in any direction. It reached but a very good precision for the axial direction, so below 7 nanometers. And what is really completely original is that it's really uniform. There is no variation in comparison to all the other techniques that vary along the capture range. I've shown you images up to 30 microns. We recently tested 40 microns, and we also tested brain slices, and it works also. And where we are currently working very hard in order to be able to observe with Christian all this rescue on this island is to be able, one very important point is to observe multiple proteins, because we do need that for all the biological applications. So be able to apply techniques, what we call spectral demixing, be able to observe 2 dye like Alexa 647 and Alexa 680 in the same setups in order to be able to co-localize these 2 information at the same time. So we need again to modify a bit the detection system. And we're also working on a dual objective configuration to have a precision that will be fully isotropic this time. And then also among the idea we have in mind, I mean, this concept could be extended also to orientation. So I just want to conclude on acknowledge all the people that did the work. So mainly it was the PhD thesis of Pierre that we co-supervised with Christian, that was funded by Paris-Saclay. And then now Abigail is working on this project. We've been also supported by all these students within the group, and Max is also working on that. And we've been also supported by Abelite for some software and some equipment. This is a strong collaboration also with Institut Langevin, with Emmanuel Faure and Surabi. And here you have all the paper patents and all the people that very kindly allow this to be possible. And I thank you for your attention. And we're ready for question, I think.
Hi, Sandrine. Hi, Christian. I mean, really impressive, the 3D resolution, particularly you get. Coming back to the first part of the biological questions on this kind of like crossing of microtubules. Yes. So you were wondering what's going on then. So then relating that into a 3D problem. So often, like when these microtubules come close to each other, is something like a problem of crowding. So I was wondering if you can comment like on this in terms of the imaging, if you get fluorophores that get close to each other, in Z, can you actually like, are there any limits where you can still get info, or at some point it's becoming a bit blurry?
Yes, I think actually yes, the special position of microtubules with respect to each other in Z, if I understand. Okay, so this is not accessible usually, because when we look at microtubules, we don't know which one is over the other one. But thanks to this modlock technique, we now could hope to identify which is the microtubule that crosses the other one and the reverse too.
Yeah, so just when there was then the modlock data on the microtubules at the end, I had the feeling, because I had in mind when you had your first part of the presentation, you could see the crossing very nicely. But in those particular images, I had the feeling that the fluorescence was a bit lower, precisely where you had the crossing.
Oh, so we probably have to re-display crossing. I mean, with the performances we have, we should definitely be able to, because it's like below 7 nanometers, so there should be no ambiguity or where is the top one and the lower one. So we can reinvestigate crossing, but I think there is no reason where we shouldn't be able to see these two different positions.
The only limitation will be the size of the crossing, of course, because the number of molecules that will be able to emit at that in this location is of course limited. But an interesting possibility is to envision that crossings are the minimum overlap of 2 microtubules, but if the angle changes and then some contact extends along the microtubule body, we could also answer to these questions.
So this was really very impressive, both in the biology and the technique. But I'm not sure if you said it, but I missed it. I guess now with a new imaging technique, you should be able to see these islands that appear, but you didn't show them, did you? Do we have now these very high resolution images of the islands? And maybe a related question to what Joost asked also is, I don't think you showed us the dynamics. So is this something that you're going to do of the growing microtubules and shrinking and so on? Is this like is this possible technically, and is it something you're going to do?
Well, to see the growing, we need to have something that is compatible with on live.
Yes, we should be coupling, for example, some turf imaging with the super resolution then, and to switch from one technique which can record some dynamics and then to the other technique on the same field.
But why can't we record dynamics with the new technique?
So here for now we are working on fixed cells mainly, so we can do single particle tracking if you want, but another option would be, we recently received a dye that is blinking in live cells, so we need this blinking so to do the localization. So depending on this dye that would work in a living cells, we could try to see the trace and have the 3D information. But the problem is that they're much lower in terms of emissions, so we probably won't be as precise, but that's something we need to try.
Yeah, and the second drawback is that, of course, to all the difficulty in the observation and the prediction of the location of Gtp Islands comes from the fact that you can't use the antibody to have some live recording of this, because it interacts with all the soluble tubulin in a GTP bound form that is present elsewhere. So this is why we needed to develop other tools, just like the clip-170 tool I showed you before, to precisely identify where possibly the GTP islands will lie and where we can try to catch them. Yeah.
I was very intrigued by the mitochondrial imaging that you did. So I was wondering which mitochondrial membrane you are actually targeting, and if it's possible.
I think it was
to possibly look into the space in between.
So I barely hear you. We labeled Tom 22. It was a question. I didn't hear the end of the question.
Yeah, which multiple membrane did you target?
Ah,
And
Tom
is it
22.
possible to target
22.
both and to look in between.
Mm. Yeah.
So there's a question via Zoom. So it's a question about acetylation of the microtubules. So it's more. So I just read the question. I suppose the curvature due to the acetylation has to occur on only one side of the microtubule.
Yes.
Do you have any ideas about how the TAT will detect the side of the microtubule?
We don't know. As far as I know, there has been some demonstration that can enter the microtubule by the ends, especially near the endocytic vesicles, because it's highly concentrated there. Regarding lateral openings, in fact, it's quite difficult. We already tried to correlate the presence of acetylation stretches with the presence of Gtp Islands. It's a bad correlation, you know. So this is why also we are wondering whether that could, at least in part, operate from the outside of the microtubule to acetylate tubulin, because the loop of the alpha tubulin that holds the lysine 40 is quite flexible, and maybe it could be accessible from a location between neighboring subunits. Thank you again.
Thank you.
Welcome.
Monsieur Algan, vous pouvez faire un test de micro?
Oui, bonjour, vous m'entendez?
Super. Écoutez, c'est à vous. Allez-y.
D'accord. Est-ce que j'ai accès au défilement des diapositives?
Malheureusement, non.
D'accord.
Il a fallu que vous fassiez un partage d'écran.
D'accord, c'est pas grave. Je vous demanderai de changer. Okay, thanks for the DIM ELICIT for giving me the opportunity to present my research. Next slide, please. Diapositive suivante, s'il vous plaît. Okay, perfect. So the topic I'm developing is a workflow from immunopurification to macrofluidic chip analysis for alpha-synuclein to develop an early diagnostic of Parkinson's disease. Parkinson's disease is a long-term degenerative disorder, and usually when clinical diagnostic is made, 50 to 70% of the dopamine neurons are destroyed. There's also the use of biological biomarkers as a diagnostic, but like the identification of oligomers, but is still a late diagnostic. The incriminated protein is alpha-synuclein, and this protein is in equilibrium with 3 conformers, and their abundance is related to the healthy form. When stress is occurring, like oxidative stress, the abundances of this form is changing. So we can observe that the pathological form can have a different pattern, and then that could be viewed as a structural biomarkers for early detection of Parkinson's disease. Next slide, please. Diapositive suivante, s'il vous plaît. So we developed a methodology like with from immunopurification to capture alpha-synuclein, and we made the proof of concept that there's no change in conformer abundances. Then we are using a size exclusion chromatography to change the buffer to be compatible with mass spectrometry. We're going to make an OD measurement of the protein concentration because the abundances of the conformers depend on protein concentration. And then we will record ion mobility and mass spectrometry data. That will be the point to quantify the abundances of conformers. We made a proof of concept in that case that there's a relationship of changes in conformer abundances and pathological forms. The chip is depicted here with an IP module where the antigen to antibody complex is captured with magnetic beads carrying protein AG. And then we have the size exclusion chromatographic module at low pressure. And we have the OD measurement and the coaxial gas and the direct electrospray ionization. I'd like to thank you for your attention and my colleagues again for the support of this work. Thanks for your attention. C'est bon.
C'est bon? Ouais. Et du coup, ça marche? Okay. Okay, so... Okay. Alors... Ah, mais il faut couper lui alors. Okay, so thanks for the opportunity to present my work. So in the lab, we study female gamete. Oops, sorry. So we study female gamete morphogenesis and how the oocyte acquires the ability to sustain embryonic development after fertilization. And so this developmental potential is particularly relevant for medically assisted procreation. The success rate of this procedure is still low, only 20%, and represent a burden for couples due to heavy treatments and high risk of miscarriage. So therefore, embryologists are looking for new biomarker of oocyte developmental potential to select the most promising ones for in vitro fertilization and increase the success rate.
And in the lab, recent results have established a link between oocyte cortical tension and developmental potential. So extra stiff oocyte or extra soft oocyte exhibit deleterious phenotype that prevent their further development. So this suggests that oocyte cortical tension and more generally mechanical properties might represent a promising biomarker of oocyte developmental potential for selection. However, current methods to evaluate mechanical properties of oocyte are time-consuming, invasive, and expensive, which limits their use in a medical setting. So the aim of the project is to develop a user-friendly microfluidic platform to evaluate mechanical properties of oocyte as a predictive biomarker of their developmental potential for medically assisted procreation. So the project combines technological development with the design of the microfluidic device, biological objective to establish the most effective parameters to score oocytes, and clinical application to adapt the protocol to human oocyte and clinical routines. So during the project, I developed the microfluidic device adapted to oocyte handling. It is based on a microconstriction through which the oocyte is forced by a control flow. Then I adapt the protocol to Collège de France. And I did the first test using already characterized mouse model of extra soft oocyte. And as you can see, control and extra soft oocyte can be partially discriminated by their entry time in the constriction. So I'm now developing a better mechanical modeling of oocyte deformation through the constriction to refine this measurement. I'm also improving the microfluidic device to ensure oocyte recovery after the measurement. And this development will allow the first test on human oocyte through already established partnership at Hospice Civil de Lyon. And at the end of the project, we aim to have the first proof of concept for the use of this microfluidic mechanical measurement to better select oocyte before in vitro fertilization. And so I would like to thank the partner of the project, the team of Marie Militaire at Collège de France, and also Clément Campio at Université d'Evry, and people who helped me, and the funding.
Hello everyone. So today I will present my work. So lab-on-chip with Gmo Sensor for early diagnosis. So the development of rapid and sensitive early diagnosis techniques is a real challenge for fields such as health, environment, and defense. This is why the goal of my PhD is to develop a lab-on-chip based on magnetic sensor called GN magnetoresistant sensor for the detection of biological objects one by one, for example, cells or bacteria in very small quantities. But first, what is a GMS sensor? So a GMS sensor allows detecting very small variation of magnetic field by measuring resistance variation. And our sensor consists of two ferromagnetic layers separated by a conductive but non-magnetic layers. And so one of these two ferromagnetic layers has a fixed magnetization, while the other is left free and orientated according to the external magnetic field. And this stacking results of two opposite configurations, so parallel or anti-parallel magnetization. And as you can see here in here you can see here in the sensitivity curve, we can observe the two opposite configurations, and between them a linear variation of the resistance according to the external magnetic field. So it's in this area that the magnetic field is measured. And to finish with our sensor, the special geometry in York shape, as you can see here, is designed to have an axis of easy magnetization for the free layer. But how we can detect biological objects which are naturally non-magnetic? For that, it's necessary to mark them beforehand with magnetic bead functionalized with antibody directed against the biological target of interest. And so thus all the samples are studied using our new detection device here. Includes the new pattern biochip inserted in an homogeneous permanent magnet where the magnetic field magnetizes the beads that mark the biological object. And then the dipole field of each magnetic object is detected simultaneously and coincidentally by Gmo sensor located on both sides of the microfluidic channel. And for the first time with this new biochip, it's possible to determine the flow height of the object, the number of particles per detected object, and their magnetic moment. So it's possible to differentiate biological objects from the aggregates of beads causing false positive, sorry. So thanks this new device and new biochip, we were able to significantly reduce the number of false positives and determine a new sensitivity 10 to 30 times higher than that we obtained with ELISA test or with the first prototype with a single set of Gmo sensor under the microfluidic channel. And we made also this lab on sheet transportable compared to ELISA test and flow cytometry, for example. And the next step of this project is to reduce as much as possible the aggregation of bead with a custom-made beads and to fully validate the cells model. And after to perform early diagnosis test with a bacteria model. So we already published some patent and publication on this project. And thank you for your attention and thank you to my female team and thank you to DIM ELICIT.
Good morning, everyone. Let's start. Okay, so the title of my PhD project is C. elegans development perturbed in space and time using microfluidics. But before going into my project, let's take the development of a frog as an example. The frog, as well as all the cold-blooded animals, are sensitive to temperature. And basically, higher is the temperature, higher is the developmental rate. But at the end of the development, when the frog, for instance, reaches the adult stage, the outcome is the same. And so it raises the following question: how are tissues and organs kept in synchrony during the development? We decided to address this question in a quantitative manner by studying C. elegans as a model organism that you can see here. This is a round worm. I mean, C. elegans is a round worm which is transparent at all stages of life. And for the project, we want to study the gene expression of developmental genes and the cell division pattern in the skin stem cells that you can see here in green on the illustration, and they are placed all along the anteroposterior axis of the worm. And regarding the experiment, we want to perform this long-term live imaging experiment. So first, for instance, we want to place C. elegans at constant temperature. Then during the next developmental stage, we will place the worm in a linear temperature gradient along its anteroposterior axis. And the idea is to perturb the synchrony of the skin stem cells. And basically, in the warmer part, the worm will develop faster than on the cold part. And then we will, during the next developmental stage, place C. elegans in a constant temperature again and see if the worm is able or not to recover from the perturbation. But we need to fabricate the device to generate the linear temperature gradient along the anteroposterior axis of the worm. And at this stage of development, the worm is only a few hundred microns long. So this is a real technological challenge. So the basic idea is to develop a new microfluidic device to immobilize the worm in a trap channel that you can see here. And the specific pattern of transparent microheaters, the input voltage that we will apply will allow us to get the linear temperature gradient by the Joule effect. And here, few results that we already got. So you can see the gold and the ITU resistors on top of a cover slip. Here is a PDMS chip properly positioned for the worm to face the linear temperature perturbation. So the worm will be immobilized close to the resistors that you can barely see. And a last image to show that there is no optical distortion effect regarding the heating system. So when there is a ITU resistors or not, there is no disadvantage regarding the optics. And so through live imaging, mathematical modeling, genetics, we want to basically dissect the molecular mechanism that ensures the synchrony of the skin stem cells during the development of C. elegans. That's it. Thank you for your attention.
Hello, everybody. My name is Feng Liang, a PhD student in microfluidic group of BNS. Today, my project named the AutoStem Lab, which is for automatic differentiation of human-induced pluripotent stem cells, iPS cells. The iPS cells can be differentiated into any cell types, but the differentiation process is cumbersome. You have to exchange the medium every day. You have to prepare a lot of medium with different reagents, and all the jobs are labor consuming and time consuming. So if you want to differentiate the neuron, you have to do this job for more than 3 months. So here we propose a AutoStem Lab. We want to let the machine to free your hands. I still remember the COVID-19 came last year. We have to stay at home and stop all the experiments. At that time, if we have the automatic differentiation, we can still get the results after the confinement. So we enhance the automatic process. And we also found that the automatic differentiate neuron networks are better than the manual one, because when you exchange the medium, you have to take in and take out the cells from the incubators. So this is not good for the neural network formations. Not only the neuron, the epithelium and endothelium tissue, the lung tissue and cardiac tissue, they all make sense in our AutoStem Lab. And our operation is quite simple. We see the iPS cells on the patch and then insert them into the microfluidic chips. And after plug and connect to the instance, we just push the start button and then wait for the result. We have built several configurations to fit the demands. We cooperated with closely with the mesobiotech company, and they provide stable and standard components, so we can focus on optimizing the differentiation protocol. And then next I want to give an example. We differentiate the human cardiac tissue to study the effect of the spike protein, which is from the coronavirus. We saw that a high dose of the spike protein will combine with the angiotensin II receptors, which will cause the sacrum damage and break the beating rhythm. And we believe that the AutoStem lab can make people have easy access to the stem cells. Thank you for listening.
Bon. Il y a Elodie qui veut présenter. Je vais t'aider.
Yeah, so we had the flash talks in microfluidics. These all are projects that were funded by the DIM and started like a few months or years ago. I'm very pleased to see some results that some prototypes have been built, that the collaborations with the biology staff started, and also noticing that for most of these projects that were presented, intellectual property has been created, patent application, and what we hope at the end of the day that this will be the basis for tech transfer or startup creation once the proof of the biology derived from this prototype will be obtained. So thanks everyone for these flash talks about this project. And now we're going to have a longer talk about microfluidics with Charles Barraud, who's going to present us what he's been doing with spheroids over the past few years and what type of biological application can be achieved.
Okay, thank you very much. So actually, we're going to do the presentation together with Hélène Delnery from Institut Curie. I am Charles Barraud from École Polytechnique and Institut Pasteur. So thank you very much for the opportunity to speak today, to present what we've been doing for a few years now. Maybe the first thing to do is that what I'm going to show you is work that has taken place over many years. It's a very slowly maturing project that started maybe more than 10 years ago. And in this project, we have really the contributions of different actors that have brought very, very different know-how and skills to the project. The project started at École Polytechnique a long time ago, as I said, maybe around 2010 or so. And then with the help of DIM ELICIT and other funding agencies that I will list at the end, the microfluidics was transferred to Institut Pasteur, where we've been doing more biologically related work. And then with the help of Hélène, we've been applying this to actually screening biologically and medically relevant samples. So this is kind of the general context. So I'm sorry if you've seen this slide maybe 50 times before, but the basic idea of microfluidics for me is to go from something that you can do with large and manual systems to something that you can integrate into something very miniaturized and integrated. And this exists already in the form of multi-well plates, and this is what people use very, very frequently right now. But then the question is, if you shrink down the multi-well plate, so rather than having 100 wells, if you have 10,000 wells, then you can ask different types of questions, and you can try to use this miniaturization at the same time, the integration of all of the operations on the same device to ask new types of questions. And this is kind of the philosophy that we're trying to develop in my lab. And you probably know this, but these chips that I'm showing you, you can already buy them. They already exist. So why am I talking to you about them? Well, in my lab, what we've been working on for the last few years is to replace these microfabricated chambers with droplets. And these droplets then can act as individual microreactors in which you can perform many, many operations in parallel. So we're going to work with, so what we would like to do is to really develop these microfluidic techniques for doing three-dimensional cell culture. And why three-dimensional cell culture? Because traditional cell culture methods work with cells that are sitting on the surface of a plastic dish. And that doesn't represent very well what you and me look like. We're not sitting on a plastic dish. So what has been emerging more and more recently is that cells, they adapt to their environment, both mechanically and biologically. And so what we would really like to do is to create a three-dimensional cell culture format where cells are only interacting with other cells. So the archetype for this type of system is a three-dimensional cell culture. Classically, these are spheroids. And the way that spheroids are made is that you can disperse a bunch of cells in a liquid droplet, and that's hanging on the bottom of a plate. And then by gravity, the cells will sediment to the bottom. And then when they meet each other, they start interacting biologically, and they start biologically forming the three-dimensional tissue, which then becomes the spheroid. And something that's very important about these spheroids is that these are not just an aggregate of cells. The cells in the spheroid are organized in a way that corresponds to their phenotypes, and they actually communicate with each other both mechanically, chemically, through contact, and also through secreted molecules. So spheroids are a classical system. More and more people are talking about organoids, but from my point of view as a technologist or a physicist, it's the same thing. It's just a ball of cells, but then it depends what types of cells you put inside them. And these spheroid chips actually, so the spheroid systems actually exist already as well commercially. These are systems that you at one point you were able to buy them in the US from Michigan, and then in Europe from a Swiss company in Sphero. And you can actually buy these plates and pipette your cells inside them and make the spheroids and study them. Then the question is, how can we do better than this? How can we use microfluidics to push the ability to culture and manipulate spheroids much further? So what we're going to show you today, Ellen and I are going to participate, both of us, in this presentation, where I will show the 3D culture in droplet microfluidics initially. And then we will show you how we've been using this platform with the help of the DIM ELICIT to screen drug combinations along with STGQA. Right. So, so the basic idea that emerged a long time ago now is that if you take droplets and you squeeze them between two surfaces, then you deform the droplets, and this has an energy cost. So the droplet, if you just take a droplet and you put it on a surface, then it wants to be spherical. This is to minimize its surface energy. So if you take droplets and you squeeze them between two surfaces, this has an energy cost. And if you then provide a little hole in which the droplet could actually release this confinement, then the droplets will preferentially go into this hole and stay there. So this is very, very classical physics. The surprising bit is that it works extremely well in microfluidics. So what we've done in the lab is that we make now these channels with different heights. And then if you put cells inside of these droplets, then what will happen is that as the droplet reaches the location of the hole, it goes into the hole. And then by gravity, the cells sediment to the bottom. And this does exactly the same as I showed you before. They start interacting with each other. If you choose the size of the droplet correctly and the type of cells and so on and so forth, then they can make, in principle, a spheroid. And the way that this works in practice is that we make a microfluidic chip that fits on a microscope slide. So you see this is 1 cent. So it's a small chip. And on this chip, we have 3 regions. We have a droplet production region that allows us to make the droplets using standard microfluidic processes. And then we have a region where we sort the droplets. We don't sort them, we distribute them laterally. And then a region where we trap the droplets. And then this is the region that we're going to use for most of the experiment. So this is a little movie to show you how the device works. In this movie, we're looking at this little hole here. And this is the hole where we inject the cell suspension. So what you see on this image is the interface between an oil on the outside and an aqueous solution on the inside. And the aqueous solution, so this is the interface, and the aqueous solution contains little cells that you're going to be able to see. And this is a movie in real time. So here we go, 3, 2, 1, go. And what you see is that we make the droplets, and they're nicely monodispersed from the beginning. And then as they are transported, they reach this area where they meet what these V-shaped grooves in the ceiling of the chip. And these V-shaped grooves, we call them rails, they basically allow you to distribute the droplets laterally so that the whole chip, you get drops everywhere in the chip. Otherwise, you would get them only in the center. And then when they reach these little hexagons, the hexagons are grooves in the bottom of the chip. And this is exactly what I showed you in the picture earlier. The droplets are squeezed in the regions between the hexagons, and then when they meet the hexagons,
they release their confinement, and so they go into the little holes. And what you can see is that the cells are free to flow inside of the droplets when the flow is moving. So this is just a liquid droplet where the cells are moving around. So this was in real time. So as you see, in about 1 minute, we were able to fill, a quarter of the chip. So in about 5 minutes, we filled the whole chip. And now, once the chip is filled, then we just stop the flow, and by gravity, the cells sediment. And as they sediment, they start to meet each other. And now this is a time-lapse movie. So what you're going to see is not in real time, but over about 12 hours or so. So if we take an image every 20 minutes, what we see is that the cells start to create junctions between themselves. And then there's a magic moment right now where the interface becomes purely spherical. So I like this movie. I'm going to show it again. There's a few things that you can see here. The first thing you can see is that cells begin initially pretty round, and then it becomes a spheroid that's round, and the cells are spread on top of each other. So you see these are the signatures of really membranes of the cells actually connecting together very, very precisely, and you get very nice mechanical and chemical communication between the cells at these interfaces. Another comment that's interesting to make here is that I'm a physicist, so I don't care what type of cells. So I'm mixing all kinds of different types of cells. In the lab, we've tried maybe 15 different types of cells, and it doesn't always work, but you can always find a way to make it work. So this is very generic. It can be used with stem cells or with cancer cells or with cell lines, typical cell lines. There's another little trick that we can play is that since I told you the droplets were liquid, we can then flow the oil on the outside, and this creates a flow on the inside of the droplet. And this is cute because at one point we weren't completely certain that the cells were making nice balls. So by actually just flowing the fluid, we can turn the spheroids around, and you can see that it's a really isotropic 3D cell culture. So it's a spheroid or an organoid. Right. So this is what we did a long time ago already. I mean, these papers were published in 2020, I think. So what can you do? So you can culture the cells in these devices, but then what is important? So I'm skipping a lot of technical steps, but you can actually stain the cells once they are in the device, and so you can remove then the oil and flow different solutions of antibodies. So you can fix the cells, stain them, and so on. And then by doing some imaging on these devices, what we get is that we get images that well, we don't have nice microscopes like the previous presentation. We do what we can with the microscopes we have. And so what we get is that we get 3-color imaging on each one of the spheroids, where we can resolve individual cells inside them. But we do this on hundreds of spheroids in each chip. And so what we build is what we call multiscale cytometry. So cytometry because we're measuring things on individual cells. And it's multiscale because we can relate the phenotype or the behavior of each cell with its position in the spheroid. So we know the neighbors of the cell, we know if it's on the outside or on the inside. So we can relate the structural information on the spheroid with the phenotypes of the individual cells. And all of this, we can relate also to the population level measurements. For example, either by recovering the supernatant to do ELISA, or by recovering the cells themselves to do some genetic testing. So we can build information on many different levels in the same device. Okay, I need to go faster. Sorry. So the problem with what I've showed you so far is that we're doing the same thing on all of the spheroids. What we would really like to do is to do different things on different spheroids. And the way we do this is to make these anchors with a little nose. And if I have a nose, then I can make the first droplet with a spheroid inside it, and the second droplet where I can bring a different solution. So the different solution, I can bring it just 2 minutes later, or the next day, or the next week. I have a lot of freedom on this. And so the way that this works is that first we fill the large droplet, and here we have food dye just to show that we have different contents in the different droplets. But this is an actual photograph of the device. So we have large droplets that go from yellow to blue, passing through green, and the small droplets that go from clear to dark red, passing through pink. And then when we merge them together, watch carefully, tuck, you get all the different colors. So this is to show that we can have different conditions and different droplets in the device. So we can use this now because in the first droplet, we can make a spheroid. And the second droplet, we can bring a library of droplets that contain cells or drugs or different things. And this is how we're going to do the screening. So I'll go a little bit quickly through this. In the second droplet, you can bring cells. And if the cells actually like each other, we can build tissues this way. And this is quite useful because we can then repeat this and we can make tissues with different types of cells. We can also bring a pathogen like a bacteria and see how the pathogen kills the spheroids. This is the only thing we've done so far. But the last movie that I will show is a project that I'm very, very excited about, which is where we have a cancer spheroid in the center of the droplet. So we make the cancer spheroid the first day. And then the next day, after the spheroid has been made, we bring some T cells, so immune cells that recognize the spheroid and that will activate if they touch it, and then they will start attacking the spheroid. So one thing I should say is that this spheroid is in a hydrogel, so it's not a liquid droplet anymore, but it's a fibrous hydrogel, so matrigel. So when we merge the second droplet, this is 3 hours later, we're going to do some time-lapse microscopy. And what we see is that the T cells recognize the spheroid, accumulate on it, and then they kill the cancer cells. And I think that this is the only movie showing this level of detail, both in time and in space, on the interaction between T cells and a three-dimensional structure of a spheroid. And so this is a project we're very excited about, except, well, this is not what you're going to hear about later. So we got to this and we thought we're just using cell lines. What can we do that's medically relevant? And that's when we applied for the DIM along with different partners, of whom Hélène, so she will show you the rest of the presentation.
Yeah, so the idea of building together this partnership was to bring to what Charles has presented to you, the experience that we have in the Curie Institute from the high content screening, drug screening for many years. So things that we have been doing, it's using cancer cell models, and through the library of different drugs that we can buy or we can get from chemistry groups, we put together drugs and cells, and you can see using this kind of plates, which are cell culture plates with many wells. So in which well we add a different drug and one concentration of many concentrations. And so we can see what happened, coupling the robotic systems of pipetting and adding drugs to the microscopy platform. So we can see in a very detailed way each cell, what's going on each cell in a flat biology way or in 3D. So the idea was to bring our experience from high throughput screening, from drug screening to the system, and getting things much faster and at low price, because when we test drugs, it costs a lot of money. So we use a very tiny plate, but still we are using very expensive tips, and all material it's very expensive. But we can then bring all this pipeline to the chip and see what happened to cancer cells when we add drugs. So the protocol that we started developing together was get what you've seen, the spheroid formation, the culture chip, and then adding this dimension of another chip that could send the different drugs to the cells. And the combination could then be studied and see what's happening finally for treating cells in a large scale, in a miniaturized way, and combined one drug or many drugs, because the answer we want to have in oncology is how to treat cells with a certain drug, but also how to combine what's already been done in cancer therapy many drugs and see how resistance of drugs that are known, that are done to patients can be broken. So this is the pipeline we're going to show to you. So the drug screen starts with a forming the spheroids, the cells spheroids in different containers that you see here. So you can see it's very homogeneous. Then we wait for a certain time for the spheroids to be formed. And then we come with the different drugs that we can choose. In this, I'm going to show you, we choose 2 drugs here, but you can see that each drug can be labeled with a different dye. And you can see that we can dilute the drug to do a dose response assay very quickly, and playing with the color of the mixture of the dyes and the dilution, you have different colors, and you have a system, a barcode system, very, very quickly. And here in this way, you can have a large number of spheroids that are very homogeneous inside, which is really important if you want to test different drugs. And we can load with different concentrations in a very rapid way. And only one device can give you really a lot of information that we can, if we use the plates as we do when we are doing screening, it takes much more time, of course, and much more plates to handle. And so you hit the kaleidoscope of colors than you have, which represents different concentrations of the drugs that have been mixed. So for the cell model that we choose for this project, it's a cell model. It's a cell model from Ewing sarcoma. It's a pediatric cancer. It's a very, very aggressive cancer. And why we choose this? Because, first of all, because we are working on this model at the Curie Institute. Also, it's interested some teams in Curie Institute and the hospital. And mainly because there is a very important question about chemotherapy and resistance today. So the poly therapy that is given to children is very aggressive, and there is still no cure. So the need to find new drugs very quickly and how to combine them to find a good one, it's a very translational and clinical problem, and that's what we asked here. So there is what we call a clinical regimen that is done today, in which some drugs are used. So it's a 3 therapy combined together at different time points. So for this study here, we then selected 2 drugs, so carboplatin and cisplatin and etoposide, for using testing in the system here. So it means that the interest that we have is use the system to combine drugs and to study the drug combinations. So at the first glance, you can see we culture the spheroids for many days. And you can see that for day 1 to day 11, we can't keep the spheroids on the system. But when you label them with in red here, see a dye for cell viability. So you can see that it's starting at day 5, you can increase a lot the cell death without sending any drugs. So we decided the time point could be in between D3 and D5 for the study we want to keep. So we can then grow, you see here, we can grow again using propionidine, the cell viability marker. You can grow the spheroids for a certain time. They're still growing, they keep growing. But we cannot do any experiment at a long term because the spheroids are already very, the toxicity is already very present. So you can see here, the spheroids grow in size, and after day 5, there is a stability of the growth of the size of the spheroid. They do not proliferate anymore, but also the viability starts to decrease. So we have to set up a certain point. So the first protocol we use was we start forming and seeding cells at day 1. So the next day we send the drugs, one drug or the other one, etoposide or cisplatin, and at different concentrations. So then you can see here the different concentrations of the drugs that have been using for etoposide, for instance.
And you can see that very, very nicely. You can see the size of the spheroids growing until day 3. And you can see the effect on cell proliferation with the viability marker, that very quickly the drug is acting and deforming the spheroid, and you can see form and also the viability of the cells decreasing. And for the second drug that has been sent, imaging each day, this is a very powerful thing, that the ability to see how the spheroids are behaving day per day at, according to different concentration of the drug, that you can see that cisplatin is very aggressive at 1 micromolar. Already the spheroid is completely being disrupted by the drug. And then the question we ask is about reproducibility and how robust that was. And you can see that in the microfluid chip, if you see here at day 2, we can do the dose response analysis, having a lot of data points, because in the chip you have a lot of combinations and a lot of replicates of the same concentration. So it can give you a very precise dose response curve and IC50 determination for each drug. And then you see here etoposide IC50 and cisplatin IC50 can be really precise and detect very quickly. And the question we ask also, in how could be corroborated to plate assay. And you see here that in droplets, you have very low IC50, so it has been determined. But when you go back to plates, you can see that etoposide and cisplatin have pretty much the same degree of IC50 that can be measured. So the microfluid system is really very robust for the determination. So the barcoding, now it's allow us with, as I told you in the beginning, so we have 2 drugs. So each drug is labeled with a different color, and you have a condition in which you have no drugs. So you have no color, so you have your control here. And in the kaleidoscope of colors that you have, the mixture of different concentrations will give a palette of different colors, and then you can determine where are your data points of drug alone for cisplatin and etoposide, for drug 1 and drug 2, and then the combination of the different concentrations that can be determined by the devices. You can see here now, when we plot in 2D now the viability plots using etoposide or cisplatin, and we image day by day as we did it before, and you can see very quickly that day by day you can have the viability being changed. In blue, you have viability, which is the cells are dying, and you have in yellow viability is at the top, so cells are not dying. We can see that it vary in a time course kind of assay. We have day 1 to day 5, what's happening when the cells have been treated with one drug or the other one. When you have now the addition of one drug and the other one, and this is what I think it's super powerful in this kind of the system, which is very hard to do in the plate assays, is that the fact that you can treat, as you can do in preclinical assays, one drug, and then the next day you treat the other drug, because this is how patients are treated with chemotherapy. It's in a time course assay. So this is possible using the combination of the screening in this kind of system. So we can add one drug or the other one, or we can add one drug, the next day we treated the other one, and we can do this at the different days. So what's super interesting here also is that if you treat the spheroids with etoposide, and the next day we treat with cisplatin, and we look at day 2, you can see the viability is pretty much being pushed by the cisplatin. And if we treat the drug and the other way, you treat first cisplatin and then you add etoposide, you can see that a viability is not the same when you compare both. And this is super interesting, the fact that we can play using the microfluidic devices with the sequence of treatment of different drugs. And okay, so probably you can.
Okay, so just to finish, the development of this technology was very, very long. The first time we put cells in droplets was in 2013. So you see, it's a very long time. And throughout this time, actually, we've needed support from all of the different institutions that we're affiliated with, and others that we're not actually affiliated with as well. What the DIM funding allowed us to do is to actually go beyond what we can do in our lab, and to collaborate with Hélène and with other actors. And this actually, so without this collaboration, we wouldn't have known anything about drugs, about the cell lines, about the questions that are being asked. So this was extremely important for us. But now we're at the stage where we've done what you see here is a technological proof of concept. It works in our lab. It works when experts are using it. But the real objective is to take this out to clinics, to really take patient samples and to have this being automated. And this is not something we can do in the research lab. And so this is why we founded Octobre, which is a startup that's taking this technology and actually automating it and making it basically a technology where you press one button and it gives you the answer. So this is the work now of a startup company. It's beyond what we can do in the lab. Maybe we're a little bit late, so I won't go through this. But so this is work of 12 years. So you can imagine many people have collaborated, but most of the microfluidics was done by Raphaël Tomasi, Gabriel Amselem, and Sébastien Sarr. More recent work on the cell-cell interactions was done by Gustave Rontex, Shreyans Jain, and Christelle Angeli. And then Auduran and Nadia Verti actually worked on the project with Hélène. We have many collaborators. The data shown here were from collaborations with Philippe Bousseau and Anna Kumano. And then, of course, the collaboration between Octobre and the Institut Pasteur. And then I'll let Hélène.
Yeah, so in the Curie, so from the Institute Curie, we had the team of Olivier Delattre, which is the team that worked on Ewing sarcoma in a very biological way, and also in very translational projects. And Karine Duval that gave us the sales for the work. And I have also to thank Kevin Miller, which has been recruited in the lab and have been working a lot on the development of the cell culture that help us a lot to bring the cell culture to this project. So thank you.
Thank you very much.
So we may have time for one of two questions. More
Yes,
rapid.
thank you. Yeah, nice integration. I was wondering whether you could access maybe to individual cell details like migration into the spheroids, and if this is not possible, could you collect these spheroids after the experiment, maybe to seed them and analyze migration, for example?
So what we're limited by the penetration depth of the microscopy. So what we do in the lab right now is we use spinning disk microscope. We can get very nice resolution on the outer layers, but seeing inside is more difficult. And we're applying light sheet microscope also, but that's much harder. But with the light sheet, we can have better penetration depth. So we're working on this. But I was going to ask you the opposite question, but.
Okay. You can call that a spheroid assay, right? Yes. So the naive question, in fact, what is your vision of the way how the system can be used in the clinics? Do you think that you have a patient, so you sample or you do a biopsy, and then you develop a personal treatment based on a mixture of drugs? And I guess that's a question that people ask is, are you sure that the treatment that works in the droplet will also work in the in vivo environment? And what do you think about that? Okay.
Yeah, so the idea that we had is really to go this way. And this for pediatric cancer, this is what has been has been done, and they are doing this in the kits in Heidelberg, using it is not even spheroids, but it's really using biopsies or tumor derived cells to grow them in vitro and use this as a tumor board decision. But in this case, using models that are molecularly characterized, so they know which are the drivers in terms of genetic drivers that have been looked. So they have drugs, for instance, ALK inhibitors. So they have very specific drugs that they know they're supposed to work. And then they test new drugs or even repurposing old drugs for the new therapeutic application. So we have a huge European project. We are working on that. But this is the next step idea that we have in mind. Yeah.
So thank you very much for the presentation. So I was wondering, in fact, I have two questions. The first one is, I didn't really get how you get different concentration in each of the wells. So maybe by micro droplets, or it's random. I understood that.
Yeah, it's random. Yeah.
So and the second question is, it seems that it's very well adapted for a mix of two conditions across several concentration. But do you see that as a possible extension of that work using this microchip we can find in high throughput qPCR, for instance, where they are microtube that enable to bring different drug into each of the well. And I was wondering whether you investigate into that direction.
So what I like in the technologies is when there's just one button and then the thing does it on its own. So indeed, the droplets come randomly to the chip. So we cannot control with the current technology what droplet goes where. This is why we barcode the droplets, and then by reading the barcode, then we can know what are the conditions in different droplets. So far, what we're doing is that we're using a gradient. So we're having a gradient of a certain drug, which we relate to a gradient of a certain barcode. But we don't have to do it this way. We can actually relate the intensity and the color to different solutions initially. So they could be completely independent solutions, but we use still the same barcoding. And this would work perfectly fine. There's no problem with that. But we will never be able to do very, very high, explore very, very large spaces of parameters with this. But a few hundred different droplet contents, that's no problem.
Okay, I guess we're going to move to the next talk, or
on
maybe one very last question, and then we move forward in order not to be too late.
Thanks, great talk, great tech actually. I've got one quick question, which is the barcoding, doesn't it very much interfere with your readout? So if you put in many colors to get all these different conditions, so then you are left with only few that you can read out, no, at the very end?
Yeah, so that's a good point. So yeah, you have to worry about this, but you can worry about it. Actually, you have to worry about it on a case-by-case basis. It depends what you want to read, and it depends also on the intensity of the barcode. Because what we end up doing in practice, in this picture, the secondary droplet is relatively large, but in practice we bring a small secondary droplet, so the intensity of the background signal is quite low. So yeah, you're right. You have to worry about having different fluorophores at in different droplets, but it's not a huge problem. We can deal with it by image analysis.
Okay, so thank you very much for the talk. I mean, thank you for showing all these steps leading from the first invention of the technology to the creation of the startup. And you can see, it's a long way. Like even if in technology it's much shorter than developing a new drug, it's still gonna be between 5 and 10 years between the very first moment and the creation of the startup. So we hope that this program is going to continue, that the projects you've been starting in the first mandate will lead to startup in the coming years. So now we're going to have 3 flash talks about image analysis and before the last session about the deep tech funders. So you guys going to start the talks.
Yeah, thank you everybody. So I would like to give a high appreciation for the organizer to make this symposium happens. And it's my great pleasure to present you the project of using residual network 50 deep learning for rapid droplet validation from the drop-up antibody secretion immunoassay. So I will first roughly explain you about the assay. First, we get, we have a droplet contain cells, white cells, and reagent for antibody and antigen detection. And we incubate into 2D chamber to determine whether they have relocation of fluorescence to determine whether we have antibody and antigen relocation. And what is one of the challenges of this project is how do we do image analysis to have a rapid detection whereas if you do morphological shape algorithm or threshold algorithm, we got a lot of false positive due to irregularity of bit line and cells. And what we are proposing is using deep learning of which is residual network 50 for the rapid detection. So first we have a data set of training. We have empty cell, we have single cell, and we have over than one cell. And we use this data set training to train with residual network algorithm, which is have 50 layers of calculation algorithm. And we will manage, we can get the result from the various dataset learning from 500 dataset until 68,000 dataset. From this one, we can conclude that if we have more dataset learning, we can get higher accuracy, over 99%. And we use this deep learning technique to validate one of the ongoing experiment, which is we are doing a validation of plasma cell from vaccinated patient against mRNA vaccine to check whether the cells are specific to certain RBD protein from COVID-19. So thanks again to DIM ELICIT that we were able to buy another laser and filter. So in the end, here we only have 3 different variant wild type from Wuhan, alpha, beta, and we will do the 4th one, which is delta. So once we get it, it's validated. So here, if you see where is the, okay, here we have single cell here, and here we have wild type. So we have over the time course, whether it's specific or not, we can detect by using image analysis. Thank you.
Hello, everyone. My name is Huatian Wu, PhD student at MIRCENCIE. I'm glad to be here today to present our work in neural individualization using deep learning techniques. At MIRCEN, we are interested in neurodegenerative disease, which are often characterized by the death of neurons. Here's an example of brain atrophy. We can also see this neuronal degeneration at microscopic scale with histological images. Our data came from a healthy macaque brain. Histological sections were produced, scanned with a neuron staining and scanned with a very high resolution. Here's histological section based on which we create our data sets. It has a diversity in terms of shape, size, and distribution of neurons. If you zoom in, we can see the neurons. We can then estimate neural population manually, but it's time consuming. As deep learning achieved a good performance in medical image analysis, we'd like to develop an automatic method for neural individualization in histological images using deep learning. Well, in order to have a robust deep model, it's essential to have enough data and precise annotations for training. However, manual annotations are tedious. To address this problem, we propose a pipeline for generating pixel level masks using only pinpoint centers and a region growing algorithm. Well, our masks include 3 classes: background neurons and the border of touch neurons. These masks are used as ground truths to train our deep model. Well, for this work, we tested a dozen of different networks. Most of them are units-like with a deeper encoder. The input of our model is the histological image, and the output is the semantic segmentation. The post-processing is applied to separate neurons. According to results on our validation sets, our method performed well on both detection and instance segmentation. In the future, we'd like to extend this work to study brain organization and to other cell types, which would improve our understanding of neurodegenerative brain disease and other infectious disease. Another application would be to model brain tissue at cell level to improve digital simulation and in vivo MRI imaging, as well as technology transfer to industry. Thank you for your attention, and thanks to DIM ELICIT.
So good morning, everyone. So I'm really happy to be here today, even if 3 minutes is really hard for me to talk only within 3 minutes. So we'll skip the introduction and we'll quickly introduce something fast about one of the hot topic on which we work in my lab, which is immunogenic cell death. Immunogenic cell death is a peculiar type of cell death which is triggered by some anti-cancer treatments. And I won't get into the detail, but the nice thing is that some anti-cancer treatment can stimulate your immune system to respond to cancer. And for a long time, so you can understand the clinical relevancy of finding ICD inducer among anti-cancer chemotherapeutic agents. And until now, for quite a decade, I think, we were based on the molecular signature of this immunogenic cell death. By the way, this drawing is quite old, but it's for the sake of simplicity, I selected it. But well, we based ourselves on this molecular signature, which was quite cumbersome because we needed to screen for drug that was inducing all these events. So we needed fluorescent, biosensor, and so on. That was quite cumbersome. And quite recently, we published a new paper showing that this molecular pattern in vitro using cells was strongly correlated to transcription inhibition. And so that was nice because in the end we were like, okay, that's great. So instead of screening for all this molecular feature, we can start by screening for simply transcription inhibitor. And there are several ways to do that, but they all require stainings and some cumbersome steps. And that's the reason why I decided to go towards machine learning using label-free imaging, because that's really cost efficient. We can screen really fast without the requirement of anything but the drug library. So the pipeline is quite simple, and you all know this kind of pipeline. We have a compound collection, we treat cancer cell. Then we used automated imaging. So in the past, we were using fluorescence imaging, but now we just use bright field imaging. And here is the nice stuff. To treat and analyze the image, we use a dual deep learning approach. I will get into the details right below here. And we simply identify hits. So here is a quick view of what we're doing. So we get bright field images, and we use the first fully convolutional neural network, SCNN. Here we use the UNet architecture, was taught right before, to get the semantic segmentation of our images. Then we use conventional natural morphology image analysis to just filter, clean the masks, and extract the cell of interest. For example, we just get rid of the apoptotic cells and so on. And we extract single cell images on which we run another deep learning model, but this time not a fully conventional, but a simple convolutional neural network, which is called VGG16, that allow us to classify the cell into transcription inhibition phenotype or not. And I'm not giving details right now about the training, but we train these two models here on several thousands of images to achieve an accuracy. I don't remember the AUC parameter, for example, was 0.99. So really nice accuracy. And this algorithm is working really, really well. And now we're trying to transfer this method to different technologies. And we are now applying that to flow imaging cytometry to detect the phagocytic event when we co-culture dendritic cell together with cancer cell. And it's quite promising. That's all for me. Thanks for your attention.
Thank you very much, the three of you. So for the last session of this morning, the idea was to let two scientists explain how they've been to down the entrepreneurship road. These two persons that are gonna, make a testimonial today of their adventure have been supported by DIM ELICIT to access the Deep Tech Founders training. And so the idea is that they will share. They were scientists, they were postdocs like you. So the step it takes to create a startup, how they decided to do it, what the training brought them. In a very few minutes, like just launching some ideas, but then that's the opportunity, if you want to discuss further with them, to be connected to other people like them in your field, we can do it for you. So now we're gonna try to reach to Maximilian on Zoom, if he's around. So interestingly, last time this DIM event was totally digital because of COVID, but now Maximilian has to go digital because he has COVID. Not not a very bad one, but yet positive, and he has to isolate. So not. Yeah, we're a bit early, in fact. So maybe Marina or Marion, can you reach him?
Emilien, vous m'entendez?
Je vous entends bien.
Super.
Ah, oh, pardon. Qu'est-ce qui se passe?
Ça, c'est vous, ça.
Ça doit être Marion, je crois.
Oui, c'est Marion qui se demande. Par contre, si vous allumez votre caméra, on pourra vous mettre en full screen.
D'accord. C'est Marion, elle a tout fait bugger dans mon ordinateur, là. Alors, mince, j'ai perdu Zoom. Où est-ce qu'il est? Il est là. Super. Bon, bonjour tout le monde. Vous m'entendez? Allô, allô, allô.
Tu nous entends?
Je vous entends bien. Est-ce que vous m'entendez?
Ouais, on t'entend très bien.
Super.
Alors, on va te mettre en full screen ici. C'est en cours.
Wow. Vous allez voir ma tête en énorme.
Donc, il y a une bonne trentaine de personnes que tu
Bonjour.
ne vois pas forcément.
Je ne vois personne.
Est-ce que tu me vois, moi? Coucou.
Coucou, mais je ne sais pas qui c'était. Tu es tout petit.
C'est Elodie, mais c'est super
Ah,
avec les
Elodie,
masques.
salut. Elodie, salut, pardon. En fait,
Je suis un tout petit.
Tu es en tout petit dans un tout petit icône de Zoom. Excuse-moi.
Donc, je vais te poser quelques questions pour guider la conversation. Et donc, le technicien essaie de te mettre en grand écran. Bon, là, on te voit en petit, mais je pense qu'on t'entend, donc peut-être...
Je suis pas sûr de vouloir... Oh là là. Je suis passé
voilà, ça va mieux. Donc,
ah, bah, ça y est, je te vois. Merci,
donc,
monsieur.
Voilà, ok. Donc, déjà, en fait, est-ce que tu peux nous dire qui tu étais, où tu étais il y a quelques années avant cette aventure Akemia et ce que tu faisais comme recherche?
Avant Akemia, en français ou en anglais?
Good question. What do you prefer? Like, let's do it in English. Like, then we can make sure that anyone understands. There are some non-English speakers in the audience.
Okay, very good. So before Akemia, so Akemia is 2 years old, and before Akemia, I have been a researcher for my whole life. I have a PhD in quantum mechanics from CEA at Saclay. Then I did 2 postdocs, one in Oxford, UK, one in Cambridge, UK. And then I came back to École Normale Supérieure and as a CNRS researcher, and so got the since 2013, and I led my research group at École Normale Supérieure, doing what? Statistical mechanics of the liquid phase, and second, a little bit of generative AI as applied to inventing new molecules. So I was really a researcher doing statistical physics.
So what are the key events that led you to think you may want to create a company?
So first, I had already co-founded or funded several companies before Akemia when I was a postdoc, but very, very non-ambitious, without a lot of ambition, just for fun. But when I was a researcher, I discovered that so I was not working. So Akemia is doing drug discovery. We are designing new compounds, and that's not at all what I was working on when I was a researcher. And what happened is that I discovered that a company called Schrödinger, a very good name for a company, a US-based company, was communicating on the interest of doing binding free energies, calculations of binding free energies for the sake of doing drug discovery and inventing the future of drugs. So that's when I started to get interested in that because that's what I was doing. I'm an expert in statistical mechanics to computing free energies, Gibbs free energies, Maxwell free energies, etc., etc. So I discovered there were companies leveraging this kind of calculations for drug discovery. And I looked at the technology. The technology we had developed at ENS was the same precision, but 10,000 times faster. So we thought, okay, if they sell this for hundreds of millions to large pharmaceutical companies, maybe we have something to do with our technology and to help people find better drugs and faster. So yeah, that's how all started.
And so at what moment you thought it was a good idea to get a specific training for this? And who were the people who helped you down the road of creating the project?
So first of all, I participated to how do we say un concours in English? I mean, I got a prize for the project, a startup project, which is called Startulm. And there I met several people with more business insight than me as a researcher. And they told me, and they were right, that I absolutely had to find the right co-founder, a business co-founder, and that I had to, open myself to other fields, more business field, IP, technology transfer, a lot of stuff. And so, yes, it was like obvious to go through the deep tech founder. Yeah, does it answer your question?
Yeah, and so maybe you can tell us more about your co-founders and what the team looks like now.
Okay, so in terms of co-founder, I co-founded Alchemia with Emmanuel Marciano, and she was a principal at Boston Consulting Group, BCG, so doing strategic consulting for 10 years. And she left the BCG. I left the CNRS and École Normale Supérieure to co-found Alchemia. This was exactly 2 years ago. In the meantime, and to do what? To doing drug discovery. So really we design compounds and we propose them to pharmas, and if we don't propose them to pharmas, we literally pay for the synthesis, for the testing, etc., etc. And we are now 21 people located in Paris, un boulevard Pasteur, very close to Institut Pasteur, even if we are not a spin-off of Institut Pasteur. We are 21. We raised 10 million in total, and doing a lot of stuff with pharmas and biotechs. And yeah, does it answer your question?
Yeah, I mean, just also to give a glimpse of people where the company is now. So maybe not to make it too long. Here you have postdoc, PhD students, and researchers like you in the room attending the conference here on site and online. So what can you share with those who may think they want to create a startup at some point? What type of advice would you have given to yourself 3 years ago, and would you have appreciated at that time to take the step of going into that adventure as a permanent researcher? So just to let everybody just know what's the situation. So Maximilien decided to go fully into the creation of Alchemia, the drug discovery startup, and to suspend the CNRS contract for the time being. That's one modality in which you can do this. There are others, but he decided to go down that road and to be now the CEO of the company himself. So what would you tell to your former colleagues?
Wow. So first of all, I learn every day, so I'm doing mistakes every day. So my advice today may not be a good advice. Second, it's a very intense journey. So I think it would have been a bad idea on my side, impossible, not to fully quit the CNRS to doing the job as a CEO. It's a full-time job to be a CEO, in my opinion. And also to be aware that the technology is not everything at all. It's only a small part of what is a company, and of the importance of having good co-founders. I mean, that's a journey that really, without Emmanuel and now the whole team, it would be completely impossible. We spend a lot of time together. We make a lot of decisions together, and finding the complementary, challenging co-founder with whom you can work in trust. That's one of the key learnings. Do I answer your question?
Yeah, so I think this is, in fact, an interesting testimonial, but you mentioned that you met this co-founder in an event organized by Like the tech transfer, basically, that was how it happens. Or did you look for your co-founder yourself?
No, I'm in Startulm. I met someone who became very important in academia, but who is not a co-founder, who is an ex-ENS, ex-BCG. So he made the connection with Emmanuel. So I started to network in this event. I started to network and to understand that I could not find by myself the right co-founder, the right team, etc. I had to open my chakra, as we say. Does it answer your question?
Yeah, it does. I mean, I guess that you know, the human adventure within this startup adventure is almost as important as the technology. But if you don't have a good technology, it's also not working. So
Yes,
maybe like the insisting that the combination of both make it work. And maybe Rémi, you will give us your insight about also this human aspects. Okay, it's going to be more challenging to organize this back and forth between you two online,
yes,
as I initially wanted, but maybe we can give it a try.
So, I'm really sorry for not being with you. I would have loved to do that, but I got a positive test to COVID, even if I'm vaccinated, so I could not join, and I'm sorry about it.
Yeah, and that's the reason that even we're here, and we're very happy to have you with us today. We still have A, to wear mask, B, we don't have the possibility to invite you for lunch afterwards because this is still forbidden. Sure. We're really sorry about that too, but yet we're not done with these epidemics, but it's nice to have you here. So maybe Rémi, you can introduce yourself, tell us what you did as a scientist before creating Sonio, and a bit what Sonio is doing in like a few sentences.
Okay, so it works, yes.
Vas-y.
Là, c'est bon? C'est bon? Oui. Okay. So I'm Rémi Besson. I'm the CSO, so scientific director of Sonio. Before that, I was a PhD student in applied mathematics in École Polytechnique. And after that, I was a postdoctoral researcher in Université de Paris. And so, well, from my PhD, I was working already on that subject of using artificial intelligence, well, decision tree and Bayesian networks. This is what we use today, but we will use also automatic image recognition tomorrow to detect and diagnose rare diseases in prenatal diagnostic. So based on ultrasound images. So this is what we do.
Yeah, so and how about the co-founding team of Sonio? Who are your co-founders and how did you meet them?
Yes, so I did my PhD with two PhD advisors that are co-founders of Sonio, that are mathematicians from École Polytechnique, and they were also three physicians from Necker Hospital that are the ones that formalize the problem that they had, and so they are also the co-founders. So it's already a large team of co-founders. And we have like 4 late co-founders that came 1 year ago and that were here to bring their skills to make scale the company. So there is a CTO, which is expert on all these technical aspects. And we also have people from business more. As the CEO, sure. How can I forget that? Cécile Brosset, that I met during Deep Tech Founders. So during Deep Tech Founders? Yes, so it was during, in fact, an event organized by Deep Tech Founders. It was
So initially you registered to Deep Tech Founder on your own, like from the lab, and yes. Then you met your CEO co-founders during the training in a workshop of the training?
The current CEO of Sonio, yes. We met her
okay.
with my PhD advisor that was also doing the deep tech founders.
And what were the key elements that led you all, I mean, the three of you to decide that you were the right team to create the company together?
Yeah, I think we all felt at the end of my PhD thesis that there was something to do, and that maybe the pure academic way was not the best way to make this project give, I mean, a software used in real life. I mean, we need other skills like technical skills to build that. We need a business skill to do it. So we understood that we needed to open our team and to found the company.
So how many people are they now in at Sonio?
I guess something like 20 or 30, because there is many people in freelance also. Well,
yeah.
So it's a different. But we are 20 at least full employed by Sony.
So your case is a bit different than Maximilien. You are a PhD student, so you just jumped into it at the end of your PhD. Was it something like when you started your PhD, you knew you wanted to do that, or does it come along the way?
Yeah, it comes little by little. But I knew when I start my PhD that I wanted to work in healthcare, because I wanted well, to have an impact, and that the research that we make has an impact, a realization in the real world. So I was already thinking about that, but not that it was going to be a startup.
So, I mean, the idea is now if there are students or postdocs also here, maybe you can add the connection to Rémi for further details about the adventure. So maybe to ask you both now, you know, without making any publicity, but we picked this deep tech founder training as a DIM to co-fund these trainings and help you acquire some new skill on the way to creating this startup. So can you maybe share what was the major learning, major take-home message that you got during this deep tech founder training? So before you answer, to let everybody know, this program is a program on several months where you will come with a specific project, your science, and you will try to figure out if you can make a business out of it and how. So you will have some workshops, you will have a lot of testimonials from startups, you will have some basics about how company works, you will have tons of feedbacks from entrepreneur or different type of profiles in company or IP. And so these are like trainings that will happen like once a week during several weeks. And then you have also a coach which is guiding you towards acquiring knowledge to build your project. So this happens over a semester. And typically DIM ELICIT co-funded half of the price of this training for a couple of research team, including you both. So maybe Maximilien, what was the main take-home message for this training? The biggest shock or surprise?
I mean, there are two things that pop up in my mind. The first one is the feedback and the challenge. It is not something I was used to as a researcher. I mean, you challenge the ideas, but it's very different talking about science on the whiteboard and being challenged from the business point of view, because it's much more fluffy and soft. And so having the feedback from the team, from the team of deep tech founders, was extremely useful to us. And the second thing was all the discussions with the networking with the founders, the people that, I mean, I can think of many, I mean, at least 4 talks that really enlightened me on why they were doing this journey, what to wait for during this journey, and what to expect. And yeah, learned a lot.
And so, Rémi, what can you share?
Yes, what I found really amazing with
We don't hear you.
I'm sorry, I cannot hear anything, in fact.
Hello?
I don't know if you're talking to me or... Ah.
Et là, ça va mieux?
Top.
Oula, il y a un écho maintenant.
Ça va être compliqué.
On va retester pour voir si l'écho a disparu.
Allô?
C'est bon. Et là, tu ne nous entends plus?
Ça y est, je vous entends. Désolé, vous m'aviez parlé. En fait, je savais pas s'il fallait que j'arrête. Je sais pas.
Donc, écoute, désolé de ces aspects techniques. Rémi was talking about how the deep tech founders came at the right moment where they were discussing the pacte d'associés, so like the contract to fund the company, and they were like thinking, you know, they're having to decide what to do with the people who wanted to stay in academia, also wanted to leave, and that's where we were when it stopped.
Yes, exactly. Thanks, and it was yes, a very good advice of the deep tech founders team to manage that, because it's really hard. We want them to keep motivated because we need them for the project, but we know that they can't pretend to as much as they could ask at the beginning compared to people that have jumped entirely in the startup team. So yes, that I've been of good advice on that.
So in any case, I mean, one key take-home message here might be, you know, reach out to people. I mean, you have to learn every day during this adventure. I mean, I bet you're both still learning every day, but maybe now, you know, you can be people that scientists can reach to get feedback about how the different steps went. So maybe, I mean, we can take some questions from the audience if there are some. But we are also the last obstacle between the audience and their lunch outside. So tough position in the conference. So And if there are no questions, I want, I might thank you both again. You have one question for Remy?
It's not a question. I'm sorry. It's a little bit of marketing of academia. We are we are recruiting. I mean, a lot.
Okay, fair enough. Do your advertising if you're recruiting their postdocs
Thank you
students in the room.
No. So we are recruiting PhDs and postdocs doing machine learning, biochemistry, chemistry, bioinformatics, chemoinformatics, statmex. Please do reach us, contact@academia.com or careers@academia.com, or reach me through the DIM. I mean, they know me. Elodie knows me very well, and I'd be very, very glad to answer any email, any question you may have about the position in academia, or if you want to create your startup. It's not an easy journey, and I'd be very pleased to help if I can.
Charles has a question.
Yeah, I'm not applying to academia. I was curious if the two of you could tell us a little bit about how you raised funds, and so both of the companies, I guess, are doing data analysis and computing stuff. So maybe you don't need as many funds as companies making machines. But I'm curious about how you were able to raise funds in Paris, and in this very, very competitive field of data analysis, artificial intelligence, and all this. Do you think you can grow quickly enough? Do you think you have access to enough markets to be competitive internationally?
Okay,
you want to start, me? Yeah.
We have made a first fundraising. It was on last December. So it was a small fundraising. It was in order to be able to industrialize the first product. That was what I done in my PhD. But because in my area, in healthcare, there is a strong regulatory limitation. So you have to do clinical validation. There's much money to spend on regulatory, and it's also makes the timeline pretty long. So it was the first fundraising, and we have marked the first product in May, and now we will do a second fundraising, much more higher, of something like 3-4 million, and it is for the US market and to develop new products in image recognition. Yes, it's still small, and we will grow even more with this new fundraising for images and going to US market.
But you managed to raise funds in France, not having to raise funds from elsewhere, or the US for now?
We didn't raise the money with funds for the first one.
Mm-hmm.
It was a business angel, and there are people from Canada too that were in the prenatal diagnostic world.
Okay. And what about you, Maximilien? Do you want to also comment?
Yes, I can tell you what our funding equity story, as they say in the VC world. So we funded Academia 2 years ago. 1 month later, we were only the two of us, Emmanuel and I, and the technology, and a deal with PSL, valorization, strong supporters of Academia, and so we raised 1 million with a VC called Delaya through its PSL Innovation Fund. So once again, it's like we were like feeling ourselves very comfortable in the sense that we were from PSL, we were supported by the DIM ELICIT for our training. Then we moved for the license of the technology with PSL valorization. Then we got funded 1 million by a VC called Delaya, who manages the PSL Innovation Fund. So it was quite natural. And then 18 months later, we raised with another VC, a European one, 9 million. So it was a few months ago. So that's the first part of the question, the equity story. Second part was about going global versus staying French, etc. So we are working in a drug discovery world, and this is a global field. It does not make any sense to trying to stay French or to stay European. So the next step for us, and that's part of the, is to go to the US. So we are already working with pharma companies in 3 continents, including the US. But we will open certainly an office, whether in Boston or New York, in the next few months. And so the Series B should be, I mean, somewhere in the US. I don't know. That's what I think today, but I may be wrong. But yes, you got it. Do I answer your question, Thomas?
Okay. So if there are no more questions, I mean, I thank you very much. I mean, company like you can use the DIM newsletter to share job offers. We always do that. So postdocs, PhD students, just scroll down in the newsletters, the job offer at the end of it. So that's something we can do. We will connect you with both Maximilian and Remy upon request, or to other entrepreneurs in fields more relevant to you. That's something that we will do for anyone in the network. Now that, as Marina presented before, you see we connected to a number of companies in the Paris area with whom we're maintaining good relationships. So thank you both for being there, despite your COVID test and at your busy schedule. And thank you all for attending this morning. So as I said, unfortunately, COVID, no lunch or poster session with glasses and food to pick everywhere. I'm very sorry about that. So we will have to reconvene after anyone deal with his own lunch break at 2:30 here in this same room. Thank you very much.
Bye, everybody. Thank you very much.